This recording is going to go over Chapter 4, PowerPoint A. Chapter 4 deals with tissues. And as you remember from Chapter 1, tissues are made of cells and make up organs. So at the lowest level of classification, tissues are made of cells and products of cells, and tissues come together to make up organs. As you probably know by now, the body is made of 4 basic types of tissues. And oftentimes tissues are compared to fabrics. And so just like a garment could be made of more than one type of fabric, let's say a coat could be made of wool and nylon and velvet, so organs in the body are made of more than one type of tissue. Typically, you can find all 4 types of tissue in an organ. And so the next PowerPoint will show you a couple of organs and how the different tissues are put together to make these organs. And this PowerPoint next is a summary of the different types of tissues, connective, epithelial, muscle, and nervous tissue, and what their functions is and their location. So what we're going to do with in this PowerPoint is go over the 4 types of tissues and the subdivisions within each type. So first we take the tissue, the type of tissue, and we go over the general characteristics of the tissue. The general characteristics means these are the characteristics found in every subcategory of the tissue. Every type of epithelial tissue has the same general characteristics. So we're going to start with epithelial tissue. And epithelial tissue is found covering and lining the surface of hollow organs. So when we describe the location of this tissue, we typically use the words lining surfaces. Epithelial tissue is often referred to as the carpet of the body because it carpets the surface of hollow organs and the outer surfaces of the body. Epithelial tissue also makes glands. So most glands in the body are going to be made of what's called glandular epithelial tissue. And this makes sense because epithelial tissue is made of cells. Cells are going to create the substances that are going to be secreted by glands. So one of the characteristics of epithelial tissue is that it's made of cells and that these cells have polarity, meaning that they are facing different environments. Just like a carpet is sitting on the carpet pad and a carpet is facing 2 environments, the bottom environment, which will be the carpet pad, and the top environment will be the surface. So epithelial tissues face 2 environments. The apical surface of epithelial tissue is facing the surface of the body, the exterior of the body, or the lumen of hollow organs. So let me explain these a little bit. And I can see that Michelle joined us. Again, if you have a question, just feel free to interrupt and ask questions at any time. Okay, so the lumen of a hollow organ refers to the interior of an organ. A hollow organ will be, for example, the stomach, the intestines, the blood vessels, the uterus, the urinary bladder. Okay, I can't think of any other. But any organ that has a space inside will be a hollow organ. So the lumen is going to be the interior of the organ. So that would be the lumen of the organ. So the epithelial tissue is going to be lining the lumen of the organ, like a carpet. It's going to be carpeting the lumen of the organ. I'm going to make a thicker tissue so we can see it. The apical surface of the tissue, right here, this will be the apical surface, right there, in red. Okay, the apical surface of the tissue is going to be facing the lumen of the organ. Okay, the space within the organ. The basal surface of the tissue is going to be sitting out here. That will be the basal surface. of the tissue, and it is typically sitting on another tissue, which is nearly every single time connective tissue. Okay. So the first thing to note is epithelial tissue has two surfaces: the outermost surface, which is the apical surface, the bottom surface, which is the basal surface. It is also good to note that the apical surface of the tissue oftentimes exhibits modifications. So the apical surface, so this is the tissue right here, made of cells. So this is a layer of epithelial tissue, and these are the little cells that make up the tissue. The nuclei of the cells are the red dots. The basal surface of the tissue is down here, sitting on connective tissue. Okay. The top surface, the apical surface, oftentimes will exhibit modifications like cilia, which are going to be little protein fibers extended onto the surface, or microvilli, which are folds of the cell membrane. So we will note macrovilla and cilia on epithelial tissue if the tissue has those modifications. So here's a depiction of epithelial tissue up here. Note a few things. First of all, epithelial tissue will be this tissue right there. But the tissue that is below epithelial tissue is always connective tissue. Okay, so that's the first thing to note. So the apical surface of the tissue is going to be up here, which is facing a surface. So this will be apical side. The basal side is down here. That will be the basal surface, which is sitting on a little membrane, which you can see here, those little lines right there. Let me go ahead and erase, and I can show you again. Okay, these little lines right here will be the basal membrane. And basal membrane is like a glue made of sticky proteins that are going to stick the tissue to the tissue underneath, which is connective tissue. So that's the basal surface right there. Now also notice that the tissue is made of cells, and that's a characteristic of epithelial tissue. It is made of cells, next to cells, on top of cells, etc. Notice that what catches your eye in the tissue are not the entire cell necessarily, but the nucleus of the cell, which typically stains darker, and that's what really you can see. The rest of the cell is a little bit lighter. In this particular instance, on the apical surface, there is cilia, which can be visible. Okay. So again, there's another depiction of epithelial tissue. This is another depiction of epithelial tissue. And let me note something here. In nearly every single micrograph of a tissue, you're going to have depicted more than one tissue, because it's impossible to collect a specimen of only one tissue. Remember, these tissues all come together, making up organs. So in this particular picture, at the surface of the specimen right here, that's the epithelial tissue. And notice that epithelial tissue is lining the surface. This will be the lumen of the organ from which this tissue came from. Okay. So that will be a surface. The tissue underneath is connective tissue. Again, always underneath epithelial tissue, we're going to have connective tissue. The cells of the epithelial tissue, you can see them there. They're kind of tall little cells with a nuclei in the middle. Okay. So one of the characteristics of epithelial tissue is that it's made of cells. Cells next to cells, on top of cells, and these cells are glued together by proteins, different types of proteins, but the most important one, a group of proteins that connect these cells to each other, are called desmosomes, and they're depicted right here. So these green things with these fibers going back and forth between one cell and the other are going to be desmosomes. So desmosomes glue the cells together and keep the whole tissue as a unit. Also notice another form of desmosomes down here, which are attaching the cell to the basement membrane on the basal surface of the cell. So this is the basal surface of the cell. This will be the apical surface of the cell. So remember desmosomes as the proteins that keep these epithelial cells together. So going on to characteristics of epithelial tissue. Epithelial tissue is made of close packed cells. There's no room for anything in between cells. They're held together by proteins called desmosomes. There are a few others like tight junctions that do the same work, but desmosomes are the most important ones. It is always supported by connective tissue. And then this right here is a very, very important characteristic. Epithelial tissue is avascular. Avascular, every time you see an A in front of a medical term, means no. So this means no vascular, which means no blood vessels. Epithelial tissue has no blood vessels, which represents a problem, because if you have a tissue made of living cells, those cells need to receive nutrients, they need to receive oxygen, they need to have a way to remove waste. Those are the functions of blood vessels, but epithelial tissue has no blood vessels that reach them, which means that in order to keep these cells alive, underneath epithelial tissue is connective tissue. And near all connective tissue is vascular. So the blood vessels of connective tissue are going to bring the nutrients and the gases, the oxygen, which are going to diffuse from the blood vessels and reach the epithelial tissue and feed it and keep it alive. So that's an important concept. That's the reason why connective tissue is described as supporting epithelial tissue above it. So connective tissue is found underneath epithelial tissue because connective tissue has the blood vessels that can keep epithelial tissue above alive. Another point about epithelial tissue being avascular is that if you have a tissue that is lining the surface of organs and the outer surface of the body, you want that tissue to be avascular because those are the surfaces that could be in contact with foreign substances, and you don't want those foreign substances to have easy access to transport blood vessels that could take those substances all over the body. So the tissue that is lining the surface of the body or the surface of hollow organs has to be avascular. Think of the skin. If you scratch the skin lightly, no blood comes out. You have to scratch deeper past the epithelial tissue into the connective tissue underneath before you produce some blood. Okay. So it's a protective feature, the fact that epithelial tissue is avascular. Now, epithelial tissue is described as avascular, which means all of these structures that epithelial tissue makes are going to be avascular. So that's another important point about the avascularity of epithelial tissue. Now, notice that it says that epithelial tissue is innervated, which means that nervous tissue reaches the epithelial tissue. So even though it has no blood vessels, it does have receptors, for example. Okay, nervous system receptors. The tissue has a high rate of regeneration, which means that it can repair itself and grow. All right, so it says that that's just a discussion question to make a point. In this as you go through these tissues, what you need to know for lecture is going to be the location where the tissues are found and the functions of the tissue and the descriptions of the tissue, including any special cells or characteristics that identify the tissue. For lab, you need to know the location of the tissue, any special characteristics that identify the tissue, like different types of cells or protein fibers or cilia, etc. But you don't need to know the function for lab. Okay. So, for example, as you go through examples of structures in the body, you should be able to identify structures where epithelial tissue are found. So if it says oil glands, well, we already said that most glands are made of epithelial tissue, so oil glands will obviously be made of epithelial tissue or glands. It says membranes lining the inside of bones. Okay, lining is the key word. So membranes are made of epithelial tissue. Okay. So now, one of the reasons why we start tissues by explaining the classifications of epithelial tissue is because epithelial tissue is it's classified in a very logical manner. Epithelial tissue is classified based on two criteria. One of the criteria, I'm kind of getting ahead of myself. I'll come back to the PowerPoint that I'm skipping in just a minute. So epithelial tissue is classified based on two criteria. One criteria is the number of cells, of layers of cells that is making up the tissue. If the tissue has only one layer of cells, we call it simple epithelium. If the tissue has more than one layer of cells, we call it stratified epithelium. So that would be the first criteria. And you can see here one little layer of flat cells. So this will be simple epithelium. Here you can see many layers of cells, more than one. So that will be a stratified epithelium. The second criteria to classify epithelial tissue is going to be the shape of the cells. And there are three choices of shapes. The cell can be squamous, meaning flat. And a squamous cell typically has a little flat nuclei in the middle. The cell can be a square or cuboidal, and cuboidal cells typically have a round oval circular nuclei. The cell could also be columnar, meaning taller than wider, and in this instance, the nuclei is typically oval. So those are the three possible shapes. So if you look at this epithelial tissue here, for example, you're going to see, first of all, this is epithelial tissue. Okay. Then notice in the middle, right here, this is not epithelial tissue. This is connective tissue. And then down here, you have another layer of epithelial tissue. Okay. This is a space, so this will be a surface. Here, this will be another surface right there. So if you had to classify, let me keep going like this. This is another layer of epithelial tissue. Okay. And this right here is connective tissue. All right. So as I'm explaining the organization, what would you say this tissue is, this epithelial tissue? Would you say this epithelial tissue is simple or is stratified? What would you say? Anybody? Take a guess. No? Remember that simple epithelial tissue is made of one layer of cells. Stratified epithelial tissue is made of more than one layer. Here you see one layer, one layer, one layer. You can see here the little cells. These are little cuboidal cells. They're taller than they are wider. Okay. Underneath is connective tissue, so this is just one layer of cells. So this is simple epithelial tissue, simple columnar epithelial tissue to be precise. For this tissue to be, let me go ahead and draw something. For if this tissue was stratified, we would have another layer of cells on top of the first layer. That would make it stratified, okay? But that's not what we have. So this is simple columnar epithelium. This tissue right here, again, we have, let me go ahead and identify the layers. Right here is epithelial tissue. This is the lumen of the organ. This is epithelial tissue. These right here in the middle, that is connective tissue right there. So even if it's just a thin little layer of connective tissue, that is connective tissue nonetheless. And then here we have another layer of epithelial tissue. The previous statement was, okay, yeah, of course. Okay, let me go back. Let me go back to, okay, so somebody wants me to explain again this, why is this simple epithelial tissue? Okay, so remember, simple epithelial tissue is made of one layer of cells. Stratified is made of more than one layer of cells. So when you look at this tissue, and let me rephrase these, it has to be adjacent layers. The layers have to be touching each other. The minute that you have another tissue in between, now you don't have a single continuous layer. So a stratified epithelium has to be a single continuous layer of cells. So let me go back to be stratified. Okay. So let me go back to this tissue right here, and let me identify these structures. This right here is space, the lumen of the organ. from here to here, we have epithelial tissue. This right here, the next tissue underneath, this is not epithelial tissue. This is connective tissue. Okay. Then we have another layer of epithelial tissue right there. And both layers are made of columnar cells. So this is one columnar cell, that's another one. These cells are pretty tall. And the little dark circle in the middle is the nuclei of the cell. So these little tall cells, one next to the other, like little soldiers lined up, are one layer of epithelial tissue. This is another layer of epithelial tissue right here. Again, lumen, this is surface, and here's yet another layer of epithelial tissue, little cells lined up next to each other. This right here is, again, connective tissue, which is always found supporting epithelial tissue. So this is the reason why this is simple epithelium, is because we have one layer of columnar cells, connective tissue, one layer of columnar cells, surface, one layer of columnar cells. If this tissue was stratified, what we would have is at least two layers of columnar cells. So there will be another layer on top of this one. Okay, that's what would make it stratified. Does that clarify it a little bit better? I hope. I'm going to take that as a yes. Okay, I'm going to take that as a yes. So let me go for this one now. Again, this one is made of, okay, good. So this one again, let me go ahead and identify the layers. And I'm going to kind of ignore this because it's a little bit confusing what this is up here. So I'm going to go for the area that is very, very clear. This right here is epithelial tissue, and it's one layer. Okay. This is lumen space. This is, again, another layer of epithelial tissue. Now, this layer, I'm going to make it in red, even though it's already red. This layer right here in between, even though it's thin, it still counts. This is connective tissue. Okay? Which means that the layers of epithelial tissue are single layers. This is, again, simple epithelial tissue. There's another epithelial tissue layer. So this tells you that because we have only one layer of cells, one layer of cells, one layer of cells, separated by connective tissue, which will make this tissue here connective also, actually, even though it looks a little bit weird. This is simple epithelium. Now, as far as the shape is actually not bad. These are actually square cells. The little round nucleus is the giveaway that these are square cells. So this is going to be simple cuboidal epithelium. Okay. Next picture here is a little bit more challenging because we cannot really see the shape of the cell. If these were square cells or cuboidal cells, we'll see more of a geometrical shape to them. Okay, so we'll see more of the shape of the cell. These are not, which tells us that this is actually flat cells. So this tile floor appearance is typical of squamous cells. Again, it's it's difficult to see whether this is simple or is stratified. So that's why the question only asks for the shape of the cell. So this is squamous epithelial tissue. This one right here, okay, so take a look at this one. This is a tube that was cut in a cross section. So this was a tube, and the tube is made of one layer of little square cells. So those are the one layer of square cells. Okay, making this tube. There's another tube right here. The pink tissue in between is connective tissue. So that's what tells you this is simple epithelial tissue is because we only have one layer of cells. So this little tube is made by this one layer of cuboidal cells. Okay. So this is simple cuboidal epithelium. This right here, okay, this is easy to see, one layer of cells, and this is a space, and this is a space. You could also say it's a surface. And the cells, you can barely see the little nuclei in between. They're very little cells, flat cells. So this is simple squamous epithelial tissue. Okay. So this is, again, the way we classify epithelial tissue is going to be based on how many layers and then based on the shape of cells. So before I go on, let me go ahead and summarize all the tissues all the epithelial tissues we're going to be covering. So we're going to be covering simple epithelial tissues with squamous cells, with cuboidal cells, and with columnar cells. So we're going to be covering simple squamous epithelium, simple cuboidal epithelium, simple columnar epithelium. When it comes to stratified epithelium, cells can be squamous, it could be columnar, or they could be cuboidal. So stratified is squamous, stratified columnar epithelium, or stratified cuboidal epithelium. Now, what we're going to do in this class, however, is we're not going to talk about stratified columnar epithelium because it is a rare form of epithelium, or stratified cuboidal because, again, it's not very common. So we're going to concentrate on these four. However, we are going to add two more to this list. To the stratified tissue, we're going to add, to the simple columnar tissue, we're going to add pseudostratified, which is a type of simple columnar tissue. Pseudostratified columnar epithelium. So a modification of the simple columnar epithelium is pseudostratified columnar epithelium. On the stratified group, we're going to add another stratified tissue that doesn't quite follow the rules, called transitional. And it doesn't follow the rules because it does not have squamous, columnar, or cuboidal cells. It actually has round cells. So we eliminate two, we add two. Okay. So those are the tissues we'll be covering in the chapter for epithelial tissues. Okay. So the first epithelial tissue we're going to cover is simple squamous epithelial tissue. This is a very important tissue. We're going to spend a little bit of time on it because it's very important. We're not going to spend that much time on most of the other epithelial tissues, but we do spend a lot of time on simple squamous epithelial tissue. First of all, this tissue is the thinnest tissue in the body because it is made of one layer of little flat cells. You could not get any thinner than these. Essentially, what you have is what looks like just a little line of cells. Okay. In this particular picture, the simple squamous epithelium depicted is found in the lungs, more specifically in the alveoli of the lungs, which is the air sacs of the lungs. So when we breathe, oxygen is collected in these little air sacs made of simple squamous epithelial tissue. This very thin tissue has a very important function. It allows what we call the diffusion of substances between one environment and another environment. So in between these alveoli, there are blood vessels, and they're tiny and they're difficult to see, but there's one right here, for example. These blood vessels are called capillaries. Capillaries are the smallest of blood vessels. And capillaries are actually made of the same tissue as alveoli. They're made of a single layer of simple squamous epithelial tissue. So these are the thinnest possible of all capillaries. So what you have in the lungs with this very, very thin tissue is one structure made of simple squamous epithelial tissue, and adjacent to it, another structure made of simple squamous epithelial tissue, which is the alveoli. Because both structures have the thinnest tissue in the body, both of them are so thin that they're going to allow for the movement of tiny molecules like oxygen from one of these structures into the other structure. So oxygen can diffuse from alveoli onto the capillaries only because the tissues making up both structures are the thinnest tissue of the body, simple squamous epithelial tissue. Okay. By the same token, carbon dioxide, CO2, can diffuse from the capillaries into the alveoli. So the function of simple squamous epithelium in this instance is allows for the diffusion of gas because it's such a thin tissue. Okay. So that's the function of epithelial tissue. In other instances, it's going to line surfaces, creating a very thin little tile floor, a smooth tile floor that may be moist, a moist surface. And we'll see that here in just a second. All right, so the location, one of the locations of simple squamous epithelium will be the alveoli of the lungs, and lining the surface of all blood vessels, including capillaries. I'm not going to worry about this PowerPoint. Just showing you another place where we find epithelial, simple squamous epithelial tissue. I do want to talk about this PowerPoint. The one on the right is showing you simple squamous epithelial tissue from the lungs, from alveoli. This one right here, on the other hand, is showing you what happens when simple squamous epithelial tissue creates a surface, like in the case of the tile floor that lines blood vessels, for example. Okay, so this is what it will look like. It looks a very smooth lining that will allow for things like cells to roll around within these surfaces. So let's talk about the other two locations of simple squamous epithelial tissue. One of the locations is lining, here goes that word again, the surface of vessels, lymphatic vessels, blood vessels, and even the heart. Okay. Simple squamous epithelial tissue that lines the surface of vessels, especially blood vessels, is given a special name. It is called endothelium. What this does is that it economizes words. So instead of having to say simple squamous epithelial tissue lining blood vessels, you just say endothelium. Okay. So this PowerPoint is showing you endothelium. So here is a blood vessel. This is not a capillary. This is a thicker blood vessel. It has muscle in the middle. It has connective tissue on the outside. But take a look at the inside of the blood vessel. This red carpet right here is endothelium. So instead of having to say this little red carpet here is the simple squamous epithelial tissue that lines blood vessels, you can just say this is the endothelium. This is a micrograph of a blood vessel, a cross-section of the blood vessel, and these little black lines right here are actually the simple squamous epithelial tissue that lines blood vessels. This is the thelium. Okay, so very, very, very important term. Definitely know it. Count on seeing this term on the test. Endocardium is in blue, just mentioned there. Actually, this simple squamous epithelial tissue lining the heart is typically called endocardium. Sometimes you hear it called endothelium. Endocardium is a better term for it. So definitely know endothelium. Now, down here, back to this PowerPoint, the other special location for simple squamous epithelium is mesothelium. Mesothelium is the simple squamous epithelial tissue found in serous membranes, lining serous membranes. Remember serous membranes from chapter 1. These were the double layer membranes that cover the inside of cavities and the outside of organs, the pericardium, the pleura, and the peritoneum. So let me show you what it looks like. This is a serous membrane here, and mesothelium, the simple squamous epithelium that lines the surface of the serosa is right there. So instead of having to say simple squamous epithelium found lining the serous membranes, you can just say mesothelium. Okay. So again, count on these two terms, endothelium and mesothelium, appearing in the test. You definitely have to know them. Incidentally, if you've ever heard of the term mesothelioma, oma will be a tumor. This will be a tumor of the mesothelium, and this is a form of cancer that sometimes forms in the pleura, in the pleural serosa, in the serosa of the lungs, on people who had been exposed to asbestos. These little cells right here, the mesothelium, begins to grow and form tumors, cancer cells. So that's what mesothelioma is. Okay, moving on. Again, we're not going to spend this much time on the other epithelial tissue. This is just epithelial. A simple squamous epithelium is so special that does deserve that much attention. All right, the next one is simple cuboidal. Easy one to recognize, really, because let me show you here. No other tissue looks like this, hopefully. This is one layer of little square cells. So there's the little one layer. This is the lumen of the structure. The little cells, this is the nucleus of the cell. There's one little cell, there's another one, there's another one, there's another one. You get the picture. The pink in between is connective tissue. This is another little structure here, etc. The location of the tissue is found making kidney tubules, which are little microscopic tubules found in the kidneys. The function of the tissue, back to this PowerPoint, it says it has a secretory and function, that's location, there we go, secretion and absorption. So the function of the tissue is to secrete substances onto the lumen and to absorb substances from the lumen onto blood vessels. So this right here is a little blood vessel. Remember that the kidneys, there's another blood vessel right here. The function of the kidneys is to filter blood. So the filtrate fluid is circulating inside this lumen. Some of those substances need to be reabsorbed, moved past the little cells back into the blood. Other substances need to be moved from the blood into the lumen of these little tubules. So the function of these little cells is to allow for this movement of things back and forth, which we call secretion and absorption. Okay. So that takes care of simple cuboidal epithelium. Moving on to simple columnar epithelium. So now we're going to have cells that are taller than they are wider, and these little tall cells that look like columns typically have an elongated oval-shaped nuclei, which this picture is not depicted, but it's supposed to have little oval nuclei. Location. This tissue is found lining most of the gastrointestinal tract. It's starting at the stomach, right here, and then continuing on to the small intestines and the large intestines. So most of the surface of these tube will be lined with simple columnar epithelium. The function of the tissue is the same as the function of the simple cuboidal epithelium. It's going to be absorption and secretion, which makes a lot of sense, because if this is the tissue found lining the GI tract, the function of the organs is to digest food, to absorb the nutrients, to secrete digestive enzymes. Okay, so these little cells are going to secrete digestive enzymes that are going to break up the food into small particles. Those small components are going to go through the little cells and be absorbed by blood vessels in the connective tissue underneath this epithelial tissue. So that's why the function is absorption and secretion. Here's another depiction of the tissue. Again, you have these little columnar cells right here. This is nice oval nuclei in this case. This little space is the lumen. So that's the lumen of the organ. More lumen here. That's a little bit better. Lumen right there. Okay. This is connective tissue. Okay. And just incidentally, you know this is connective tissue because you have cells, cells, cells. The cells are not necessarily close to each other. There's stuff in between. That's characteristic of connective tissue. So that's connective tissue. That's not epithelial tissue. Let me go back to the epithelial tissue itself. And again, these are the little cells. These are columnar cells. Simple columnar epithelium. Look, notice these funny looking blue things right there. Those are actually cells. There are specialized cells that are found in the simple columnar epithelium. They're called goblet cells, and goblet cells secrete mucus. You definitely have to know them. And they will secrete mucus onto the surface that will help with the process of digestion. And notice also this blue line here on the apical surface of the epithelial cells. These that blue line is actually the microvilli of these cells. So these little columnar cells on the apical surface, let me get and make a few of them. So this will be the basal surface, which is that area right there. On the apical surface, that's the nucleus, the little membrane is going to wrinkle. So it's going to create folds like that. And those folds, the purpose of the folds is to increase surface area for absorption. So food particles can attach to the surface and be absorbed. The more surface, the more food particles can be absorbed. So that's the function of microvilli, and that's why these cells have microvilli. The other form of columnar epithelium, simple columnar epithelium, is a very special little tissue called pseudostratified ciliated columnar epithelium. Pseudo means false. Stratified will mean many layers, but this is false layers, which means that the tissue itself is not stratified, it's actually simple. So this is actually simple epithelial tissue, but looks stratified. The cells are columnar, so you can see here the cells, tall cells. And the reason, it's another little cell here, cells down here. The reason why this tissue looks stratified is because the cells are not all the same size. Some of the cells are taller than others. And so it gives the illusion of being stratified because the nuclei are at different levels. I mean, another one here. So you can see the nuclei at all different levels, and that gives the illusion of being stratified. A characteristic of this tissue is that it's ciliated, meaning it has these protein fibers that stick out of the cell membrane. The tissue is found lining the surface of the upper respiratory tract. Okay, that means nasal cavity. Then the tissue disappears for a little while in the throat and is picked up again in the voice box, in the windpipe called the trachea, and the bronchi that bring the air into the lungs. So lining those surfaces of the respiratory tract, most of the respiratory tract surfaces will be pseudostratified ciliated columnar epithelium, which makes a lot of sense because this epithelium is ciliated. So let me go ahead and show you a couple of things here. So every time you take a breath in, you're breathing with that air all kinds of particles, dust, pollen, bacteria. The respiratory tract is the #1 portal of entry of microorganisms. So you're breathing in all this stuff that needs to be trapped, pushed out of the body. And that's part of the function of the cilia. The cilia is going to trap these substances. And then the cilia will begin to move beat, and it beats away from the body, sweeping these substances out of the body. Something else that's going to help with the elimination of these foreign particles will be goblet cell. So there's a little goblet cell right there. So the stratified ciliated columnar epithelium has goblet cells, just like simple columnar epithelium. Goblet cells are going to secrete mucus, and mucus is sticky, so it's going to help to stick the particles onto the cilia, and when the cilia beat and move, it helps the cilia sweep substances out of the body. So you should know the location where these tissues are. The function is described as propulsion of mucus by ciliary action. Okay. This reminds me of something else. When it comes to the function and location of these tissues, which you do have to know, under the study guides in the Module 1 PowerPoints, there is a little exercise that gives you all of the tissues and all of the locations and asks you to match location with tissue. And then there's the answer to the exercise. There is another exercise that gives you all the tissues and the functions of the tissues and asks you to match function to tissue, and then again gives you the answers. So if you're having problems or if you need to practice this information, it might not be a bad idea to refer to those two exercises. Okay, so there's another depiction of pseudostratified ciliated columnar epithelium, which you can see here is coming from the trachea. Just to note, this down here is connective tissue, which we always find underneath epithelial tissue. This right here is the actual epithelial tissue. Okay, so that takes care of simple epithelial tissue. What we have left will be the stratified epithelial tissue. And we're going to see two types of a stratified epithelium. We're going to see a stratified squamous epithelium and transitional epithelium. So we're going to tackle the stratified squamous first. First thing to note is that a stratified epithelial tissue is going to be named after the shape of the cells on top of the tissue, not the cells at the bottom. So this whole pink area here is epithelial tissue. Notice that the cells on top are flat. The cells at the bottom, you can see the round nuclei indicating the cells have more of a square shape to them. And as the cells are moved up to the surface, they're going to flatten. So this is what happens to stratified squamous epithelium. The tissue is called squamous because of the cells at the top. The cells at the bottom are young, actively dividing cells. They have a nice round nuclei. As the cells divide, the daughter cells are being pushed to the surface of the tissue. And as they moved onto the surface, they are moved farther and farther away from the connective tissue underneath. So this is connective tissue underneath, supporting the epithelial tissue on top. Now remember, the source of oxygen and nutrients for the epithelial tissue is right here, where the connective tissue is. The further the cell moves from the connective tissue, the harder it is for the cell to attain oxygen and nutrients. So these cells are going to begin to die. So they begin to flatten and begin to get older and eventually will die. Nonetheless, the ones at the top, you can still see the nucleus, even though some of them may be dead cells. Good way to differentiate between stratified epithelium or simple epithelium with connective tissue. Typically, the way to differentiate, oftentimes, especially when you have connective tissue all together, is that the epithelial tissue is going to be at the surface. So you look for a surface, and you know that the tissue right at the surface is going to be epithelial tissue. Now, the question is, how do you know how far the epithelial tissue goes? Why isn't this just, let me see here real quick. Why isn't this just that portion epithelial tissue and then everything else connective tissue? Well, this is why. When you look at the, when this is the surface of this is the part of the tissue that's adjacent to the surface. Okay, so that's definitely epithelial, hopefully, definitely. And now these cells underneath here, you can see the nuclei, and so you can see how close together the nuclei are located to each other, suggesting that the cells are next to each other. So you can see how that ends right about here. And the cells down here, this is a nuclei. That's a nuclei, meaning there's a cell here, there's another cell here, there's another cell here, another cell here, another cell here, cell, 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 cell. The cells are not touching each other. In between cells, you have this stuff. See that stuff right there? All that is the stuff in between cells. That's typical of connective tissue. That's what tells you this is connective tissue. Okay, is that you see stuff between cells. When it comes to connective tissue is cellular, meaning one cell next to another cell, next to another cell, next to another cell. So that's what tells them apart. And I'm going to say also, may not always work, but oftentimes the colors will be different. And that will be another giveaway. Then you can move on here to right there. Yeah, take a look at these right here, this picture. This is epithelial tissue. Notice there's a change in color back down here. Okay, so when you see a change in color, that typically indicates also a change in tissue, even if it's just a change in the shade of the color. In this case, you can see cells separated by this pinkish matrix. That's connective tissue. Whereas here, and I know you can, how come this other purplish stuff in between is not matrix? Well, it's not. The nuclei of the cells are too close together. Let me show you a couple more pictures right here. Again, changing color. All these right here is epithelial tissue. Now we're changing color, that's connective tissue. So look for both things. Look for changing color, which indicates change in tissue, even if it's just a change in the shade of the color. Another one here. I'm kind of jumping, but I'm going to come back to where we were before in just a second. In this instance, this is the epithelial tissue. Everything down here was connective tissue. You can see a change in shade and a change in structure, if you will. You can see almost fibers all throughout here. That's connective tissue. And no, it is not easy. Okay, a lot of this is learned more than anything, but hopefully those tips help a little bit to distinguish between epithelial and connective tissue. Okay, does that help a little bit, hopefully? And I'll continue to point out, will the basal layer of epithelial tissue always be more dense in cells and nucleus than nucleus? Not quite sure what you mean, but let's see. Let me go back to the question real quick. Will the basal layer of epithelial tissue always be more dense in cells and nucleus? The basal layer, okay, if you mean that if it is more dense in the, if it is more dense on the, okay, let me go, nuclear, this always have more nuclear. Oh, yeah, okay, got you. I understand what you're saying. Yes, the answer is yes. Now I understand what you're saying. Yeah. Okay, yes, the basal layer of the tissue will have more cells, absolutely, because these are younger cells. And these younger cells are actively dividing, and so that's why there are more of them. By the time you leave these lower layers here, these cells are not dividing anymore. Okay, so that's what you kind of see, older, less clustered cells. So now I understand the question. Absolutely, yes, that's exactly what is happening. Yeah, okay, good. All right, that was a good question, actually. All right, so that is your stratified squamous. Now, this is the deal with the oh, let me go back before I move on. Function. The function is easy. The function of the stratified squamous is what we called protection from mechanical abrasion. So protection, because the tissue is thick, so that it can withstand being scratched. Also, the top cells are dead, old, or dying, so who cares if they are sloughed off? There are younger cells underneath that will take their place. So that takes care of the function and also the location, because the location will be any place subject to mechanical abrasion. For example, okay, the mouth is subject to calibration. When we eat, we have food in the solid food in the mouth that kind of scratches the inside. It's not in here, but the rectus, the last part of the large intestine, will be subject to the waste scratching its surface. So in that last area, you will find these stratified squamous epithelium. Another one is the vagina. In intercourse is submechanical abrasion. The other one would be the surface of the skin, the epidermis of the skin. It's always being scratched, mechanical abrasion. The other one right here is the esophagus, and the esophagus is this tube right here, which brings food from the throat to the stomach. And as the food passes through the tube, it scratches the surface of the tube. So again, any place subject to mechanical abrasion will have stratified squamous epithelium. Now, let's just complicate things a little bit. There are two types of stratified squamous epithelium. One type has what we call keratinized cells, and the other type has non-keratinized cells. Okay, so keratinized cells are cells that have made a protein called keratin, which is tough and dry. Think of nails. Nails are made of keratin, fingernails. Think of hair. Hair is made of keratinized cells. Think of calluses on the skin are made of keratinized cells. So keratinized stratified squamous epithelium is the stratified squamous epithelium found on the epidermis of the skin, the top layers of the skin. So what happens to this tissue is that, let me go ahead and show you a picture. There we go. Let me give me a second, see if I can have a better picture. I thought I had, that's not a better, no, that's not a better picture. No, okay, let me go back, sorry. Okay, I'll make do with this one. All right, so this whole area is the stratified squamous epithelial tissue. But notice that here on the top, this layer looks like just fibers, doesn't even look like cells. You don't even see a nucleus anywhere in there. These are the dead keratinized cells. So what happens in the skin is that the bottom layer of cells are live, young cells, which continue to divide and send the older cells towards the surface. And as these cells climb up to the surface, they begin to make keratin. They begin to be completely filled with keratin. The cell eventually dies, and what's left is this shell of a cell filled with keratin. And that's the keratinized layer. So that's what we call these keratinized stratified squamous epithelium. So the function is the same. It would be protection from mechanical abrasion. On the other hand, the other type of stratified squamous epithelium found all over the body is going to be non-keratinized, which means that the cells on the surface are not going to be filled with keratin. They're just going to be regular cells, old cells, dying cells, but live cells nonetheless, which you can still see, or at least a little while longer. You can still see the nucleus all the way to the top of the layers. Non-keratinized stratified squamous epithelium is found lining the esophagus, the mouth, the throat, rectum, anus, vagina. Function, protection from mechanical abrasion. So when you see micrographs of these kinds of tissues, first thing you look for at the top for nuclei, the little dots, okay, which are the nuclei of the cells. And that tells you, oh, there's nuclei on top. That means this is stratified, non-keratinized, stratified squamous epithelium. So this whole layer is stratified, non-keratinized, stratified squamous epithelium. Younger cells at the bottom, that's what you have, this multitude of little nuclei that gives you the darker color. And then as you move up, cells are become flatter. They're not as young. Some of them may be dying. You can see some cells here without nuclei because they're dead. However, all the way to the top, you'll still see some nuclei. On the other hand, keratinized layer is going to have fibers of keratinized dead cells on top. The cells that make keratin are called keratinocytes. And keratin is this tough protein that is dry. It is water repellent and very, very protective. So the very top layer of the skin is this keratinized tissue. So that sometimes when you scratch the skin, you get these little flakes of dead cells, keratinized dead cells. All right, we've already covered these, so make sure you know where we find keratinized stratified squamous epithelium. Okay. Another depiction of the same. This top layer are the keratinized cells. The rest are the cells that are still alive. This is connective tissue underneath. Okay, again, a layer of non-keratinized epithelium right here. This down here will be connective tissue. Notice the change in color, or at least shade of the color. It's actually a very nice picture. These little cells down here are the germ cells of epithelial tissue. Nice little young cells. You can see the cells at the top being flat, but you can still see nuclei on top. Okay, so that's the key. The key is to see nuclei at the top. Okay, last of these epithelial tissues, transitional. Very, very special tissue because it's only found in a few places. It is found in the urinary bladder and then other structures of the urinary system, the ureters and the urethra. This is a stratified tissue because you have a layer of cells. However, it doesn't quite fall under the normal classification because the cells are round. So the cells are round, and they are going to change in shape when the organ is filled. So the urinary bladder is filling with urine, and as it fills with fluid, it stretches. As it stretches, these cells have to transition in shape from round to being elongated. And that's what they're called transitional epithelial tissues, because they transition in shape. The micrograph pictures that we have of these tissue is always coming from empty bladders, and that's why the cells are going to look round. It's because the bladder was empty. Okay. Now, the function of the tissue is to stretch as the organ distends, and you should know the location. Urinary bladder is the easier one to remember. One thing about this tissue is that it is oftentimes confused with non-keratinized stratified squamous. Don't let that happen to you. Look at the top and look for the round cells. The round cells are the key. These round cells here are not the same kind of cells seen on a stratified squamous epithelium. The stratified squamous epithelium, the cells on top are flat. Okay, there you go. These cells on top here are flat. Okay, all these little cells are flat. Okay, so just don't let the fact that they're both layers of cells confuse you. Okay, again, more depiction of the same, and look at the layers, the cells on top, which are clearly round cells with a round nuclei. Okay, let's see. The next portion of the chapter looks at glandular epithelium, and I know we're going over time, but I'm going to go ahead and finish epithelial tissue. Let's see. So glandular epithelium, most glands are made of epithelial tissue, and there are different types of glands. We talked about endocrine glands in chapter 1, which secrete hormones onto the blood. Exocrine glands secrete their substances on surfaces. The surface can be the skin, it can be the lumen of an organ. So, for example, the exocrine glands that secrete digestive enzymes will be exocrine glands. The glands that secrete saliva are exocrine glands. Okay. Second here. Most glands are going to be multicellular. A few glands are unicellular. Goblet cells are considered glands, which are unicellular glands. Okay, so that's fairly, hopefully, straightforward. So you should know the different types of glands, exocrine glands and endocrine glands, and then most glands are multicellular. You should identify goblet cells as unicellular exocrine glands. Okay. Now, the other thing that's important, and that's just a little depiction of little goblet cells there. The other part that's important is we're going to pretty much ignore this PowerPoint. We're going to go over it. Don't worry about it. This is important. Okay, these are the 3 different ways in which glands can secrete their product. And the 3 different modes of secretion are merocrine, apocrine, and holocrine. Merocrine glands make their little substance and spit it out to the outside by a process called exocytosis. So we don't call it spit it out, we call it they deliver it by exocytosis. Okay. Examples of merocrine glands include sweat glands, salivary glands, the pancreas. And just as a little note, remember the pancreas belongs to two organ systems, digestive system. And so the exocrine function of the pancreas is to secrete digestive enzymes through a merocrine mode of secretion. The other function of the pancreas is endocrine. It secretes hormones. Okay, and hormones are secreted into the blood. They don't belong to any of these categories right here. So just a little aside there. All right. So merocrine glands, and let me go back to the previous PowerPoint to show you the merocrine gland over here, right here, making the little product and secreting it to the outside. Next mode of secretion is apocrine, and what these glands do is they pinch off. They release their little cytoplasm. So little pieces of the cytoplasm are pinched off, and that's what's secreted. Examples of apocrine glands are mammary glands. So mammary glands, are going to secrete their product by pinching off little pieces of cytoplasm right there. So that's called apocrine secretions. The third and last of these glands are holocrine glands, and in this case, the entire cell is secreted. So the entire cell ruptures and dislodges from the gland, and that's the actual secretion of the gland. Sebaceous glands, oil glands, secrete their product in this manner. So you can see here in the picture the entire cell being released as the secretion. What happens to these glands is that they must be glands that are constantly regenerating. Otherwise, they will run out of cells. So you should be able to identify the 3 forms of secretion, examples of the 3 forms, and expect some type of matching in the test of these different types of secretions with a description of how they secrete their product and an example of each. Okay. So this should pretty much ends the first set of PowerPoints. This is what I want to do, though. What I like to do is I like to stick around for a little bit longer, and if you have to leave, you can leave, obviously. But I'm going to stop the recording for now and get it started again on another recording because I want to go over muscle and nervous tissue. And then I'm going to schedule another session for connective tissue, and I'll schedule it for tomorrow because I want to get this finished